Engineering Precision: Advancements in Antibody Optimization for Enhanced Therapeutic Efficacy. Narrated by Luke McLaughlin, biotech digital marketer, business developer, and life science content creator. In the rapidly evolving field of biomedical research, antibody engineering stands out as a critical technology shaping the future of therapeutics. Antibodies, with their unique ability to target specific molecules within the body, are fundamental tools in the treatment of diseases ranging from cancer to autoimmune disorders. However, the development of highly effective and safe therapeutic antibodies requires meticulous optimization. This article delves into the sophisticated biochemical techniques employed to enhance antibody properties, focusing on 5 key areas: affinity maturation, humanization, species cross-reactivity, specificity modulation, and pH-dependent binding. Each of these optimization processes is crucial for tailoring antibodies to meet specific therapeutic needs, while minimizing potential side effects. From the precise adjustments in affinity maturation that increase binding strength to the complex strategies of humanization that reduce immunogenicity in humans, these techniques represent the pinnacle of modern biotechnology. Moreover, understanding species cross-reactivity extends the utility of antibodies in preclinical testing, while specificity modulation and pH-dependent binding offer routes to target diseased cells more effectively and selectively. This article will explore how these cutting-edge technologies are not just enhancing the efficacy of antibody therapies, but are also setting new standards for precision medicine. Affinity maturation. Affinity maturation is a natural process that occurs in the immune system to improve the binding affinity of antibodies to their specific antigens. This is crucial for effective immune responses. In a therapeutic context, this process is mimicked and enhanced through various techniques. Somatic hypermutation, SHM. This involves introducing mutations in the variable V regions of antibody genes, specifically in the complementarity determining regions, CDRs, which interact directly with the antigen. These mutations can be introduced through techniques like error-prone PCR, or targeted mutagenesis. Overview of SHM. SHM primarily affects the immunoglobulin Ig genes in B cells. It occurs after antigen exposure in the germinal centers of lymphoid organs, like the spleen and lymph nodes. The mutations introduced during SHM are predominantly point mutations, which occur at a rate of approximately 10 circumflex 3 per base pair per cell generation, much higher than normal genomic mutation rates. Molecular mechanism. The molecular process of SHM can be divided into several steps: initiation, activation, re-induced cytidine deaminase, AD. The key enzyme responsible for initiating SHM is AID. AID deaminates cytosine residues in DNA to uracil, which can then pair with adenine instead of guanine during DNA replication, leading to point mutations. Targeting of AID. AID is selectively targeted to the variable regions of the immunoglobulin heavy and light chain genes. The exact mechanisms of this targeting are not fully understood, but involve transcription-dependent recruitment and possibly the specific chromatin architecture of these regions. Mutation generation. Uracil processing. The uracil in DNA can be processed in several ways, leading to different types of mutations. Uracil glycosylase, UNG, mediated base excision repair, BR. UNG removes the uracil base, creating an abasic site. This site is then processed by the DNA repair machinery, potentially leading to transitions or transversions. Mismatch repair, MMR. The MMR system recognizes the UG mismatch and removes a patch of the newly synthesized strand containing the uracil. The resulting gap is then filled in by DNA polymerase, incorporating random nucleotides and thereby introducing mutations. Replication across uracil. If the replication machinery encounters uracil during DNA replication, it can lead to incorporation of adenine opposite uracil, resulting in CG to TA transitions after the next round of replication. Regulation, enhancers and promoters. The activation and function of AID are influenced by transcriptional enhancers and promoters within the immunoglobulin loci. Transcriptional pausing. Transcriptional pausing at sites of SHM may facilitate AID action by exposing single-stranded DNA, which is a preferred substrate for AID. Mutation hotspots. SHM tends to occur more frequently at specific nucleotide motifs, known as hotspots. The WRCY, RGYW motifs, where W equals sign AT, R equals sign AG, Y equals sign CT, are particularly prone to mutations. The reason for this preference is not entirely clear, but these motifs may be more accessible to AID or more prone to DNA structural alterations that facilitate mutation. Biological outcomes of SHM. The mutations introduced by SHM can affect the antibody binding affinity in several ways. Improved fit, mutations that improve the complementarity of the antibody to the antigen can increase the binding strength. Altered specificity. Some mutations might change the specificity of the antibody, allowing it to bind to different or additional antigens. Reduced affinity. Not all mutations are beneficial. Some can reduce affinity or alter the antibody structure detrimentally. Clinical relevance. Understanding and harnessing SHM has profound implications for vaccine development and antibody engineering. By mimicking or directing SHM, scientists can develop more effective antibodies for therapeutics and vaccines, tailoring antibody responses to specific pathogens or diseases. In summary, SHM is a complex and finely regulated process that is fundamental to the immune system's ability to adapt and respond to an ever-changing array of antigens. This process exemplifies the sophistication of molecular mechanisms in biological systems and their evolutionary importance in host defense. Screening and selection. After mutations are introduced, phage display or yeast display technologies are used to identify antibodies with the highest affinity to the target antigen. This involves expressing a library of mutated antibodies on the surface of phage or yeast, and then binding them to immobilized antigen. The process of screening and selection is crucial in the field of antibody engineering and therapeutic development, particularly after somatic hypermutation, SHM, or other mutagenesis techniques have been applied to generate diverse antibody libraries. Here we will explore a detailed biochemical and molecular biology breakdown of how this process is typically conducted using display technologies such as phage display, yeast display, and mammalian display. Overview of screening and selection. Screening and selection involve identifying and isolating high-affinity antibodies from a library of variants. These libraries are often created through random or targeted mutagenesis and contain millions to billions of unique antibody sequences. The goal is to find those sequences that have the desired properties, such as high affinity, specificity to the target antigen, and suitable pharmacokinetics. Common display technologies. Each display technology has its unique characteristics and advantages. Phage display, library construction. The antibody library is cloned into phage-emit vectors, which are then used to transfect Escherichia coli. Each bacterium produces a bacteriophage particle, displaying a unique antibody on its surface. Panning process. Phages displaying antibodies are incubated with an immobilized target antigen. Non-binding phages are washed away, and those phages that bind to the antigen are eluted. The bound phages are then used to infect fresh E. coli to amplify the selected antibodies. Iterative enrichment. This process is repeated several times, typically 3 and 5 rounds, each time under increasingly stringent conditions, for example, lower antigen concentration, higher wash stringency, to enrich for phages that display antibodies with the highest affinity. Yeast display, library construction, DNA encoding. The antibody library is transformed into yeast cells, where each cell displays a different antibody variant on its surface. Binding assay. Yeast cells are incubated with a fluorescently labeled antigen. Cells that bind to the antigen can be isolated using fluorescence-activated cell sorting, FACS. Advantages: yeast display allows for real-time monitoring of binding kinetics and affinities using flow cytometry, facilitating the selection based on both affinity and expression level. Mammalian display. Library construction. Similar to yeast display, but the antibodies are displayed on the surface of mammalian cells. Selection. Mammalian display more closely mimics the human immune system, allowing for the selection of antibodies with better solubility and human-like post-translational modifications. Screening. FACS is typically used to isolate cells expressing high-affinity antibodies. Biochemical and molecular aspects of screening. Antigen preparation. The target antigen must be properly prepared and immobilized, maintaining its native conformation to ensure that selected antibodies will be effective against the natural antigen in biological contexts. Binding conditions. Conditions such as pH, salt concentration, and temperature are optimized to mimic physiological conditions or to enhance specific interactions. Labeling strategies. In some cases, antigens are fluorescently labeled to facilitate the detection of binding events, especially in flow cytometry-based methods. Selection criteria: affinity and avidity. High affinity and appropriate avidity are critical for therapeutic efficacy, particularly in oncology and autoimmune diseases. Specificity. Antibodies must be specific to their target to minimize off-target effects. Therapeutic efficacy. Selected antibodies should exhibit properties conducive to therapeutic use, such as stability, lack of aggregation, and manufacturability. Validation and characterization. After the initial selection, promising antibody candidates undergo rigorous validation. Binding affinity measurement. Techniques like surface plasmon resonance, SPR, and biolayer interferometry, BLI, are used to quantify binding kinetics. Functional assays. Antibodies are tested in cell-based assays or in vivo models to assess their biological activity. Sequence analysis. High-throughput sequencing is used to analyze the selected clones for diversity and to identify dominant sequences that may indicate particularly effective antibodies. This process of screening and selection is iterative and often involves a combination of different technologies and strategies to ensure that the most promising antibody candidates are identified and further developed into effective therapeutic agents. Iterative cycling. This process can be repeated multiple times, selecting for progressively higher affinity antibodies by using lower concentrations of antigen during each round of selection. Iterative cycling, also known as iterative selection or evolutionary cycling, is a critical process in the field of molecular biology and biotechnology, particularly in the context of optimizing biomolecules like antibodies, enzymes, or other proteins. The goal of iterative cycling is to progressively enhance the properties of these biomolecules, such as affinity, specificity, stability, or catalytic activity. This process harnesses principles of evolution, mutation, and selection, applied in a controlled laboratory setting. Overview of iterative cycling. Iterative cycling involves repeated rounds of diversification, mutation, and selection. Starting from an initial library of variants, cycles of mutation introduce new genetic diversity, and subsequent rounds of selection enrich variants with desired traits. The process is continued until the performance of the molecules meets the desired criteria. Key steps in iterative cycling. Mutation generation. Random mutagenesis. Techniques such as error-prone PCR, DNA shuffling, or mutator strains of bacteria introduce random mutations into the DNA sequences encoding the target proteins. This generates a wide variety of variants, some of which might possess improved characteristics. Site-directed mutagenesis. Specific amino acid residues are altered based on previous knowledge or predictions about their potential impact on protein function. This approach is more targeted compared to random mutagenesis. Insertions and deletions. Sometimes changes more significant than point mutations, such as insertions or deletions, are introduced to explore larger structural changes that might yield enhanced properties. Library expression. Display technologies. Variants can be expressed using phage display, yeast display, or bacterial display systems. These systems allow for the physical linkage of phenotype, the protein, with genotype, the DNA encoding the protein, which is crucial for effective selection. In vitro translation. Cell-free systems might be used for direct expression of proteins from DNA or mRNA, particularly Antibodies when working with toxic proteins or when rapid synthesis and testing are needed. Selection. Binding assays. If affinity or specificity is the target trait, techniques such as biopanning in phage display or fluorescence-activated cell sorting, or FACS, in yeast or mammalian display are used. These methods isolate variants that bind more strongly or more specifically to a target antigen or substrate. Functional screens. For enzymes, selections might be based on the conversion of substrates to products detectable via colorimetric, fluorescent, or luminescent readouts. High-throughput screening. Automation and robotics are often employed to screen large numbers of variants for desired characteristics under a variety of conditions. Analysis and iteration. Sequencing. After each selection round, leading candidates are sequenced to identify mutations that contribute to improved performance. This information can guide further mutagenesis. Characterization. Selected variants are characterized in detail using techniques such as surface plasmon resonance, SPR, enzyme kinetics, or thermal stability assays. This helps confirm their improved properties and suitability for applications. Feedback. Loop. Data from characterization studies feedback into the design of the next round of mutagenesis, aiming to further refine and enhance the properties of the molecules. Biochemical and molecular considerations. Fidelity and error rate. The balance between fidelity and error rate in mutagenesis is crucial. Too high an error rate can lead to non-functional variants, while too low an error rate might not introduce sufficient diversity to explore new functional possibilities. Fitness landscapes. Understanding the fitness landscape, how particular mutations affect function, can greatly enhance the efficiency of iterative cycling by guiding selection pressures and mutation strategies. Linkage of phenotype and genotype. Maintaining a robust link between phenotype, observable characteristics, and genotype, genetic sequence throughout the process is vital to ensure that selected improvements are accurately attributable to specific genetic changes. Applications and implications. Iterative cycling is widely used in antibody engineering to enhance affinity and specificity, in enzyme engineering to improve catalytic efficiency or substrate specificity, and in developing novel proteins with tailor-made functions for industrial, environmental, or therapeutic applications. By leveraging the power of iterative cycling, scientists can effectively evolve proteins in the lab, achieving performance characteristics that might be exceedingly rare or even unattainable through natural evolutionary processes. This method is a cornerstone of modern molecular biology and biotechnology, reflecting a sophisticated blend of evolutionary theory and high-throughput molecular techniques. Humanization, speciation. Humanization of antibodies is essential for therapeutic use in humans, particularly to reduce immunogenicity. Non-human antibodies can provoke immune responses, which can be mitigated by replacing portions of the non-human antibody with human antibody sequences, CDR grafting. The most common method where the CDRs of a non-human antibody, typically murine, are transplanted onto a human antibody framework. This preserves the antigen-binding specificity while reducing the immunogenic potential. CDR, complementarity determining region. Grafting is a pivotal technique in antibody engineering, especially in the development of therapeutic antibodies. This method involves transferring the antigen-binding regions from an antibody of one species, usually a mouse, into the framework of a human antibody. This is primarily done to reduce immunogenicity when these antibodies are used as therapeutics in humans, while preserving the antigen-binding specificity of the original antibody. Overview of CDR grafting. CDR grafting is part of the broader process of humanizing antibodies. Antibodies are composed of 2 heavy and 2 light chains, each containing variable, F, and constant, C, regions. The variable region of each chain includes 3 CDRs, which are directly involved in binding antigen. Surrounding these CDRs are framework regions, FRs, that support the CDRs structurally. In CDR grafting, the CDRs from a non-human antibody are transplanted into the framework of a human antibody. Steps in CDR grafting: identification and selection of CDRs. CDR identification. The first step involves identifying the CDRs in the donor antibody. CDRs are typically defined based on their sequence variability and location within the variable region of the antibody. The Cabot or Chothier definitions are commonly used to delineate these regions. Sequence analysis. Sequences of CDRs are analyzed and compared to ensure that they are correctly identified and suitable for grafting. Selection of framework. Human framework. Selection. A human antibody framework is chosen as the scaffold into which the CDRs will be grafted. This selection is crucial as it needs to be able to accommodate the CDRs without significant loss of binding affinity or specificity. Often, frameworks are selected based on their homology to the donor antibody's framework and their known stability and expression levels. Grafting process. Molecular cloning. The CDRs of the donor antibody are synthesized or amplified from cDNA and then cloned into the selected human antibody framework. This is typically achieved using molecular cloning techniques involving restriction enzymes and DNA ligase. Gene synthesis. Alternatively, the entire variable region sequence incorporating the human framework and grafted CDRs can be synthesized de novo, allowing for precise control over the sequence. Modification and optimization. Back mutations. Sometimes, certain residues in the human framework may need to be mutated back to the original non-human version, back mutation, to maintain the conformation of the CDRs, thereby preserving the original binding affinity. Computational modeling. Computational tools can be used to model the structure of the grafted antibody to predict and optimize interactions between the CDRs and the framework. Biochemical and molecular considerations. Protein expression. After grafting, the recombinant antibody genes are transfected into mammalian cells for expression. The expressed antibodies are then purified for further testing. Binding affinity testing. Techniques like surface plasmon resonance, SPR, enzyme-linked immunosorbent assay, ELISA, or flow cytometry are used to test the binding affinity of the grafted antibodies to their target antigens. Structural assessment. High-resolution techniques such as X-ray crystallography or cryo-electron microscopy may be employed to assess the structural integrity of the grafted antibodies and the correct positioning of CDRs. Challenges and solutions. Immunogenicity concerns. Even with human frameworks, grafted antibodies might still elicit an immune response. Further engineering, such as humanization of other parts of the antibody or modifying glycosylation patterns, may be required. Loss of affinity. Grafting CDRs into a different framework can potentially lead to a loss of binding affinity. This can be addressed through fine-tuning of the framework or additional rounds of mutation and selection, for example using phage display. Applications. CDR grafting is widely used in the development of therapeutic antibodies to treat a variety of diseases, including cancers, autoimmune diseases, and infectious diseases. By combining the specificity of non-human antibodies with the reduced immunogenicity of human antibodies, CDR grafting helps create effective and safer treatments for human use. In summary, CDR grafting is a complex but highly effective approach in antibody engineering, requiring a blend of molecular biology techniques, biochemical testing, and sometimes computational modeling to ensure success. Framework adjustment. Post-CDR grafting, some non-human framework residues may be retained if they are critical for maintaining the structure and function of the CDRs. These are identified through modeling and empirical testing. Framework adjustment in the context of antibody engineering refers to the modifications made to the framework regions, FRs, of an antibody to support the function and stability of complementarity determining regions, CDRs, after they have been grafted from one antibody, often non-human, to another, typically human. This process is critical in antibody humanization, especially to preserve or enhance antigen binding affinity and reduce immunogenicity. Overview of framework adjustment. In antibody humanization, while the primary focus is on transferring the CDRs, which are directly involved in antigen binding, maintaining the proper conformation of these CDRs is essential. The framework regions play a crucial structural role by forming the scaffold that supports the CDR loops. Adjustments to these frameworks can be necessary to accommodate the grafted CDRs properly, especially when the original donor and recipient, human, antibodies have significantly different framework sequences. Steps in framework adjustment. Selection of framework. Framework identification. The human frameworks are typically selected based on their homology to the donor antibodies frameworks and known stability. High-throughput databases of human antibodies can be queried to find the best matches. Structural compatibility. Structural analysis, possibly including computational modeling, helps to predict which human frameworks will likely support the conformational and functional integrity of the grafted CDRs. Analysis and identification of critical residues. Interaction mapping, detailed analysis of the donor antibody structure if available, or predictive modeling is used to identify key interactions between CDRs and framework residues that contribute to the stability and function of the antigen binding site. Conserved residues. Conservation of particular residues in the framework regions across different species can indicate their importance in maintaining structure and function. Engineering adjustments, Vernier zone. Adjustments, modifications often focus on the Vernier zone, which includes residues in the framework that are in close proximity to the CDRs and directly influence their positioning and flexibility. Back mutations. If certain non-human residues are critical for maintaining CDR structure or function, these can be reintroduced into the human framework. This process involves mutating specific human residues back to their non-human counterparts. Addition-deletion of residues. Occasionally, small insertions or deletions may be made to better accommodate the CDRs within the human framework. Biochemical and molecular considerations. Molecular cloning and expression. After engineering, the modified antibody genes are cloned into expression vectors and transfected into mammalian cells for antibody production. This step is crucial for assessing the function of the adjusted frameworks in a relevant system. High-resolution structural analysis. Techniques such as X-ray crystallography, NMR spectroscopy, or cryo-electron microscopy may be used to examine the detailed structure of the adjusted antibodies to confirm that the intended changes have properly supported the CDRs. Functional testing. Binding affinity and specificity. Adjusted antibodies are tested for binding affinity using techniques like surface plasmon resonance, SPR, and specificity using enzyme-linked immunosorbent assay, ELISA, or flow cytometry. Thermal stability, differential scanning calorimetry, DSC, and other thermal stability assays can assess the structural integrity of the adjusted frameworks under various conditions. Iterative optimization. Feedback loop. Based on the functional testing results, further adjustments may be necessary. This iterative process of testing and modification continues until the antibody exhibits optimal binding characteristics and stability. Clinical relevance and applications. Framework adjustment is a key step in developing therapeutic antibodies that are less likely to be rejected by the human immune system while retaining high efficacy. These antibodies can be used in a wide range of treatments, including for cancer, autoimmune diseases, and infectious diseases. In summary, framework adjustment is a sophisticated process that requires a deep understanding of antibody structure and function. It combines advanced biochemical techniques, molecular biology tools, and computational modeling to ensure that humanized antibodies retain their desirable properties for therapeutic applications. Species cross-reactivity. Optimizing antibodies for cross-species reactivity is important when developing drugs that are effective across different species, like those used in preclinical animal models. Conserved epitopes. Targeting epitopes that are conserved across species can help in developing cross-reactive antibodies. The concept of conserved epitopes is central to immunology, particularly in the fields of vaccine development, therapeutic antibody design, and diagnostic tool creation. An epitope, often referred to as an antigenic determinant, is a specific portion of a larger molecule, typically a protein or polysaccharide, that is recognized by the immune system, specifically by antibodies, B cells, or T cells. A conserved epitope is an epitope that remains unchanged across different strains of a pathogen, among different species of pathogens, or even across different proteins. Understanding conserved epitopes, definition and importance. Conservation. In the context of proteins, conserved epitopes are amino acid sequences that are preserved through evolutionary timescales within a species or across different species. These epitopes are crucial targets for broad-spectrum vaccines and therapies because they are likely important for the pathogen's survival and function. Immunological recognition. Conserved epitopes are particularly valuable in designing vaccines and antibodies that provide protection or therapeutic efficacy against multiple strains or types of a pathogen, thereby overcoming the challenge posed by genetic variability. Identification of conserved epitopes. Bioinformatics and computational analysis. Sequence alignment. Bioinformatics tools are used to align sequences of the same protein from different strains or species to identify regions of high conservation. Software like BLAST, Clustal Omega, or Muscle are commonly employed for these analyses. Conservation scoring. After alignment, each residue's conservation level is scored based on its prevalence and similarity in the aligned sequences. Highly conserved residues are those that show little to no variation across different sequences. Structural biology, crystallography, and cryo-EM. High-resolution structural data from techniques like X-ray crystallography or cryo-electron microscopy can reveal the three-dimensional context of conserved epitopes, showing how they might be presented to the immune system. Molecular modeling. Computational modeling and molecular docking can predict how conserved epitopes might interact with antibodies or receptors, helping to hypothesize their role in immune recognition. Experimental validation. Synthetic peptide studies. Peptide synthesis. Short peptides corresponding to conserved epitopes are synthesized and used to immunize animals or to bind to specific antibodies in assays like ELISA, enzyme-linked immunosorbent assay, to test their immunogenicity and binding properties. Immunological assays. B and T cell activation. The ability of conserved epitopes to activate B and T cells is tested using lymphocyte proliferation assays, cytokine production assays, and other immunological tests to assess the epitope's potential for vaccine development. Application in vaccine development. Design and optimization. Multi-epitope vaccines. Vaccines can be designed to include multiple conserved epitopes, potentially from different proteins or pathogens, to elicit a robust and broad immune response. Adjuvant selection. Appropriate adjuvants, substances that enhance the body's immune response to an antigen, are selected to maximize the immune response against the conserved epitopes. Clinical trials. Testing efficacy and safety. Vaccines containing conserved epitopes undergo rigorous clinical trials to test their safety, immunogenicity, and protective efficacy in humans. Challenges and considerations. Epitope accessibility. Conserved epitopes must be accessible to the immune system, which can be hindered by the protein's folding or post-translational modifications. Autoimmunity risks. Care must be taken to ensure that conserved epitopes do not cross-react with human proteins, potentially leading to autoimmune responses. Therapeutic antibodies and diagnostics. Broad-spectrum antibodies. Therapeutic antibodies targeting conserved epitopes can be developed for broad-spectrum activity against multiple pathogens or strains. Diagnostic tools. Conserved epitopes can be used to develop diagnostic assays that are effective across different strains or related pathogens. In summary, the study of conserved epitopes bridges molecular biology, immunology, and bioinformatics to develop effective interventions against diseases. By targeting these stable regions, researchers aim to create long-lasting, broadly protective vaccines and therapies that are less susceptible to the pitfalls of rapid pathogen evolution. Engineering framework regions. Modifying the framework regions or adjusting the CDRs can enhance the cross-reactivity without compromising the antibody's affinity or specificity. Engineering the framework regions, FRs, of antibodies involves modifying these structural parts of the antibody molecule to improve overall stability, reduce immunogenicity, or enhance other functional properties such as solubility and manufacturability. This is a sophisticated task in antibody engineering, particularly when humanizing antibodies for therapeutic use. Overview of framework regions. Framework regions are the parts of the antibody's variable domains that support the complementarity determining regions, CDRs. While CDRs are directly involved in antigen binding, the FRs are crucial for maintaining the proper orientation and conformation of the CDRs. Therefore, changes to the FRs can significantly impact the antibody's binding affinity and specificity. Rationale for framework engineering: stability and solubility, improving physical properties. Modifications in the framework can enhance the stability of antibodies under physiological conditions and increase solubility, which is crucial for therapeutic formulations. Reducing immunogenicity. Humanization. When developing therapeutic antibodies originally derived from non-human species, like mice, the framework regions are often replaced with human sequences to reduce the potential immune response in patients, enhancing expression and manufacturability. Optimizing production. Changes in framework regions can help in increasing the expression levels in host cells, commonly mammalian cells, and simplify the purification process. Methods for framework engineering. Sequence and structural analysis. Sequence alignment. Identifying the most appropriate human framework for replacing a non-human sequence involves aligning human antibody sequences to find the best match in terms of homology and maintaining the structure of the CDRs. Computational modeling. Tools like Rosetta or MOE are used to model how changes in the framework might affect the overall structure and to predict the impact on antigen binding. Molecular techniques. Site-directed mutagenesis, used to precisely alter specific amino acids in the framework regions. This technique is crucial for introducing single or multiple point mutations. Gene synthesis. Entire variable regions with optimized frameworks can be synthesized de novo, allowing for incorporation of multiple modifications simultaneously. Biochemical and biophysical characterization. Stability assessment, thermal stability, techniques such as differential scanning calorimetry, DSC, or circular dichroism, CD, are used to measure the melting temperature and stability of the engineered antibodies. Solubility tests. Solubility can be assessed under various conditions to ensure that the antibody remains soluble at concentrations required for therapeutic use. Functional testing. Binding affinity, surface plasmon resonance, SPR, and enzyme-linked immunosorbent assay, ELISA, are used to test the binding affinity of the engineered antibody to its antigen. Specificity. Ensuring that modifications have not altered the specificity of the antibody requires testing against off-target antigens. Iterative optimization, feedback loops. Iterative testing. The design-build-test cycle is a common approach in synthetic biology applied here. After initial testing, results feedback into new rounds of design and modification. High-throughput screening. Automated platforms can screen large libraries of framework variants to identify those with the best properties. Applications in therapeutic antibodies. Clinical relevance, reduced immunogenicity. By using human frameworks, the engineered antibodies are less likely to be recognized as foreign by the patient's immune system, reducing adverse reactions. Improved therapeutic profiles. Enhanced stability and solubility contribute to better pharmacokinetics and pharmacodynamics profiles of therapeutic antibodies. Challenges and future directions. Balancing stability and function. Maintaining function. While enhancing stability, it is crucial that the antibody's antigen-binding function is not compromised. Customization for specific conditions. Depending on the therapeutic application, antibodies might need to be stable under extreme conditions, such as varying pH or high temperatures. Framework engineering is a detailed and complex process requiring a multifaceted approach that combines molecular biology, bioinformatics, structural biology, and biochemical techniques. Through careful design and optimization, the engineered antibodies are made suitable for clinical applications, offering potential treatments for a wide range of diseases. Specificity modulation. Specificity modulation involves engineering antibodies to either broaden or narrow their specificity to different antigens, which is essential for therapeutic specificity and reducing off-target effects. Altering CDR loops. Specificity can be modulated by mutating amino acids in the CDRs to change the shape or charge of the antigen binding site. Altering the complementarity determining regions, CDRs, of antibodies is a fundamental technique in antibody engineering, aimed at modifying their antigen binding characteristics to improve specificity, affinity, and overall therapeutic effectiveness. CDRs are the specific parts of the antibody's variable regions that directly interact with an antigen. Each antibody contains 6 CDRs, 3 each in the light and heavy chains, and these regions are the most variable parts of the antibody molecule. Understanding CDRs. CDRs are the focal points for antigen recognition and binding. The precise sequence and conformation of amino acids within these loops determine the antigen binding specificity and affinity of the antibody. Altering these loops can drastically change the antibody's binding properties, making this a critical area of focus in antibody engineering for both therapeutic and diagnostic applications. Techniques for altering CDR loops. Molecular engineering. Site-directed mutagenesis. This technique allows for the precise substitution, deletion, or insertion of amino acids in the CDRs. Specific mutations can be designed based on theoretical predictions of how changes will affect binding interactions. Random mutagenesis. Techniques such as error-prone PCR or DNA shuffling introduce random mutations within the CDR regions, generating a diverse library of variants. This approach is useful when the specific modifications needed for improved binding are unknown. Display technologies. Phage. Phage display is a technique where a library of antibody variants with modified CDRs is displayed on the surface of bacteriophages. This method allows for the screening of a vast number of variants against a specific antigen. Yeast display. Similar to phage display, yeast display involves expressing the antibody variants on the surface of yeast cells, facilitating screening and selection based on binding properties. Screening and selection of modified CDRs. Panning. In display technologies, panning is the process of binding the displayed antibodies to immobilized antigens, washing away non-binding variants, and eluting the bound phages or yeast cells for further amplification. High-throughput screening. Techniques such as fluorescence-activated cell sorting, FACS, are used with yeast display to isolate cells that display antibodies with desired binding properties, based on fluorescence intensity indicating antigen binding. Biochemical and structural analysis, binding affinity and kinetics. Techniques like surface plasmon resonance, SPR, and biolayer interferometry, BLI, are used to measure the affinity and binding kinetics of the engineered antibodies to their antigens. Structural characterization, X-ray crystallography, NMR spectroscopy, and cryo-electron microscopy can provide detailed insights into how changes in CDR loops affect the overall structure and antigen binding site of the antibody. Optimization and iteration. Computational modeling. Before and after experimental modifications, computational tools can predict the impact of changes on antibody-antigen interactions and suggest further modifications. Iterative cycles. Based on feedback from functional assays and structural studies, additional rounds of modifications and selections can be carried out to refine antibody properties. Application in therapeutic antibody development. Therapeutic efficacy. Modified antibodies must not only bind their target antigens with high specificity and affinity, but also demonstrate the ability to elicit appropriate therapeutic responses. Clinical testing. Before clinical use, antibodies undergo rigorous preclinical and clinical testing to ensure their efficacy and safety. Challenges and considerations. Immunogenicity. Modifications in CDRs can potentially increase the immunogenicity of humanized or chimeric antibodies, requiring careful assessment and additional modifications to minimize such risks. Maintaining stability. Changes to the CDRs can affect the overall stability of the antibody. It's crucial to ensure that the modified antibodies are stable under physiological conditions. Altering CDR loops is a powerful approach in antibody engineering, enabling the creation of antibodies with tailor-made binding characteristics. This process involves a combination of molecular biology techniques, biochemical testing, and iterative optimization to develop antibodies for a wide range of therapeutic and diagnostic applications. Base-specific antibodies. These are engineered to bind two different epitopes, potentially on different antigens, which can be used to recruit immune cells to cancer cells or block two distinct pathways simultaneously. pH-dependent binding. The process of pH-dependent binding in the context of antibody engineering involves designing antibodies that change their binding affinity based on the pH of their environment. This is particularly useful in therapeutic contexts where it's beneficial for antibodies to bind to their targets under specific conditions, such as in the acidic microenvironment of tumors or in the acidic compartments of cells after endocytosis. One of the key mechanisms utilized to achieve pH-dependent binding is the use of histidine switches. Biochemical basis of pH-dependent binding: understanding pH sensitivity, protein structure, and pH. The structure and function of proteins, including antibodies, can be influenced by the pH of their environment. Changes in pH can affect hydrogen bonding and electrostatic interactions, leading to alterations in protein conformation. Histidine residues. Histidine has a unique side chain with an imidazole group, which can gain or lose a proton depending on the pH. The pKa of histidine is around 6.0, making it sensitive to physiological pH changes. When protonated or deprotonated, histidine can alter its interaction with other molecules or within the protein itself. Histidine switches in antibodies. Mechanism of histidine switches. Incorporation in CDRs. Histidine residues are strategically placed within the complementarity determining regions, CDRs, of antibodies. These residues can change their charge state with pH, influencing the antibody's affinity for its antigen. Design considerations. The placement of histidine residues is carefully designed so that at neutral pH, such as in the bloodstream, the antibody exhibits low affinity for its target, reducing off-target effects. Under acidic conditions, such as in tumor tissue or cellular endosomes, histidine residues become protonated, changing the conformation or charge distribution of the CDRs, thereby enhancing binding affinity. Engineering and testing pH-dependent antibodies. Computational modeling. Structural predictions. Before experimental validation, computational tools are used to model how inserted histidine residues might affect the antibody's structure and interaction with its antigen at different pH levels. Molecular dynamics. Simulations. These simulations can help predict the movements and conformational changes in the antibody structure in response to pH changes, guiding further engineering efforts. Experimental validation. Mutagenesis. Site-directed mutagenesis is employed to introduce histidine residues at specific positions within the antibody CDRs. Expression and purification. After engineering, antibodies are expressed in suitable systems, for example mammalian cells, and purified for testing. Binding assays. Surface plasmon resonance, SPR. SPR is used to measure the binding kinetics of the engineered antibodies at different pH levels, confirming pH-dependent behavior. ELISA. pH-dependent. ELISA assays can also be used to assess binding under various pH conditions, offering a simpler alternative to SPR. Applications of pH-dependent antibodies. Therapeutic targeting, cancer. Therapy pH-dependent antibodies can be used to target the acidic microenvironment of tumors more effectively, minimizing effects on normal cells. Targeted drug delivery. Such antibodies can be part of a drug delivery system that becomes activated only in the acidic environments, such as after internalization into cells, ensuring that the drug is released at the desired site. Diagnostic tools, biomarker detection. pH-dependent antibodies can be used to detect changes in pH associated with various diseases, serving as a diagnostic tool. Challenges and future directions, optimization challenges, fine-tuning affinity. Achieving the desired level of pH sensitivity without compromising the overall stability and function of the antibody can be challenging. Clinical efficacy. Ensuring that pH-dependent antibodies maintain their functionality and stability in the complex biological environments of the human body. Regulatory and safety considerations. Immunogenicity and safety. Any modifications to antibodies must be thoroughly tested for immunogenicity and potential adverse effects in clinical trials. In summary, pH-dependent binding via histidine switches is a sophisticated approach to creating more selective and effective therapeutic antibodies. This strategy leverages the unique properties of histidine residues to modulate antibody affinity in response to the pH environment, enhancing the specificity of antibody-antigen interactions in disease-specific contexts. Conclusion. As the field of antibody engineering progresses, the optimization strategies outlined here represent the cutting edge of biomedical innovation, promising to reshape therapeutic landscapes and usher in new paradigms of treatment. From the precision of affinity maturation and the intricacies of humanization to the tailored approaches of specificity modulation and pH-dependent binding, each technique is a testament to the scientific community's relentless pursuit of more effective and precise therapies. Affinity maturation and humanization address the fundamental need for antibodies that are both highly effective in their binding and minimally immunogenic, ensuring treatments that are both potent and well tolerated by patients. Meanwhile, the exploration into species cross-reactivity and engineering framework regions exemplifies the strategic refinement of antibodies to function across various biological contexts, enhancing the relevance and applicability of preclinical research models to human disease. Furthermore, the advances in specificity modulation and pH-dependent binding demonstrate sophisticated biochemical craftsmanship, enabling the development of antibodies that are not only selective but also adaptable to the dynamic environments within the human body. As these technologies mature, the implications for patient care are profound. Tailored antibody therapies can potentially offer solutions to previously intractable diseases, providing targeted, efficient, and less invasive alternatives to traditional treatments. Moreover, the principles learned through antibody optimization are likely to influence other areas of drug development, leading to broader innovations in biologic therapeutics. In conclusion, the advancements in antibody optimization techniques are not merely incremental improvements, but are pivotal breakthroughs that pave the way for the next generation of precision medicine. These developments promise to enhance the specificity, efficacy, and safety of antibody-based therapies, ultimately improving patient outcomes and expanding the horizons of medical treatment. As researchers continue to push the boundaries of what is possible in antibody engineering, the future holds the promise of increasingly sophisticated and personalized therapeutic options, marking a new era in the fight against disease. Stay curious, stay innovative. I'm Luke McLaughlin. If you would like to learn more about this topic, consider reading our full-length articles on www.therapeutics.com. www.biotechnologyreviews.com, or consider our podcast series on YouTube or Spotify at our Biotechnology Reviews channel. Our articles are also available on our LinkedIn newsletter, Biotechnology Reviews.